Hi, hello, namaste, good evening to all. Myself Murali.P.G under Unacademy. Here today I am going to take a YouTube class on biomolecules. Biomolecule is one of the important topic under KCET, and here regarding with the Karnataka CET 2021 aspirants, Unacademy introduced some of the important courses, and that courses are as follows. One is the plus subscription, and where you will going to get the concepts regarding the complete syllabus, MCQ, doubt clearance and mega quiz. Then you can have the unlimited access if you subscribe for this one. 12-month plan, 6-month plan and 1-month plan will be there, and use my use code or referral code, that is Murali, KCET Murali. Okay, KCET Murali is my referral code or use code, and get 10% off regarding with the price value of our subscription. That is one thing. And second one is the comprehensive batch. Here what is the speciality? Complete syllabus is covered, tips and tricks by top educators, topic wise practice questions, full length mock test will be there. So you can use this one to achieve good result in the oncoming Karnataka CET, and use the code KCET Murali and get 10% off regarding this price value. Okay, along with this comprehensive batch, I am taking the maltose batch, where we are with the detailed theory, MCQ, doubt clearance and mega quiz. It was started from 15th of December, and here myself I am taking several topics under this course. Comprehensive batch, best plan for Karnataka CET aspirants, 12-month, 6-month plan and 6-month full syllabus will be there. Dedicated doubt clearing sessions will be there. Course completion in Kannada language also. So that is the speciality. Multiple batches are launching in every month. That is what our KCET 2021 preparation. So the better things for the aspirants. Okay, daily T20 free practice test for Karnataka CET. Attempt the test for free, and here started on 2nd of November. Use it and achieve good thing from our side, and you will get whatever you want in the preparation for this Karnataka CET 2021. So with this brief introduction, along with this I am taking special classes also. If you want to join to my special class, use the app, that is Unacademy Learners app. Install it, search under Goal Karnataka CET KCET, use my name Murali.P.G or use my code KCET Murali and get the route to enter into my special class. Okay, so by this brief introduction, now I am entering towards the discussion of questions under this biomolecule, okay, chapter. So welcome to all, very good evening to all. Saurav Patil, Manohar, welcome, namaste. So the first question of this biomolecule is: enzyme and substrate complementary each other, such as. So enzyme substrate complex will be there. It is like that of what? Pencil and eraser, pen and paper, lock and key, all the above. So the right answer is C, lock and key. So enzyme substrate complex formation, it is also considered as Locke theory of enzyme substrate action. So right answer is C for the first question. So you can also give the answer, okay, through chat box after seeing the questions. Okay, so enzyme substrate complex formation is like that of lock and key. So particular lock with the particular key. So enzyme will work against particular substrate. So enzyme is with the substrate specificity or substrate with the enzyme specificity. So as a result, the reaction will be there, and that gives a free enzyme and product. Then second one, which one of the following chemical is classified as an enzyme? Which one of the following is classified as an enzyme? Triglyceride, cellulose, galactose, sucrase. Out of these four, which is the enzyme? ಇದರಲ್ಲಿ ಇಷ್ಟರಲ್ಲಿ ಯಾವುದು enzyme ಅಂತ consideration ಆಗುತ್ತೆ. So it is sucrase. Sucrase is the enzyme that will digest the sucrose particle. So always the enzyme ends with the ASE. Okay, always enzyme ends with the ASE according to the enzyme nomenclature. Okay, naming of enzyme. Third question. In the cell, digestive enzymes are mostly in which cell organelles? So digestive enzymes are present in which cell organelles? So cell organelles are of different types. One of the cell organelle with the digestive enzyme, which is that cell organelle with the digestive enzyme. Options are ribosome, vacuoles, lysosomes, mitochondria. So what is this answer for this question? So third question, what it? Third question answer is lysosomes. Ribosomes are the protein factories. This is the protein factory, and vacuole is storage area, and mitochondria is power house of the cell. So right answer for this question number third, it is lysosomes. Then fourth one, enzymes are named after their substrate adding suffix. Just now I said sucrase, isn't it? So here enzymes are named after their substrate added. Substrate is sucrose. Half portion is added with the end spelling, that is ASE suffix. It's not prefix. Prefix with that substrate. So suffix with the prefix. So prefix ಯಾವುದು ಬರುತ್ತೆ ಇಲ್ಲಿ? Fourth one, it is B, ASE. This is the prefix and this is the suffix. Suffix is the sucrase. So right answer is B for question number 4. Yes, Manohar, for the third question, it is C is the answer. Okay. Sheshika, welcome to the class. Good evening. Yes, for this fourth question, B is the right answer. Next, fifth one. The enzyme which removes hydrogen from the substrate is known as what? Removing of hydrogen particle or hydrogen part from the substrate, it is called what? It is the dehydrogenase. So dehydrogenase is the enzyme that will remove the substrate particle from the molecule, biomolecule. So that will remove the hydrogen from the substrate. So hydrogen remove ಆಗುವುದು. So okay. So the right answer for this question, fifth one, it is B. It's not D. Okay. Yeah. Yes, Shishika. It is B. Yeah, you given the right answer. B. Next question. The product is realized from which site of the enzyme? Product is realized from which site of the enzyme? So here active site. Substrate is going to be attached to the enzyme in the active site, and that product is going to have, it's not simple site, it is the active site. Product is going to be realized or released from the active site of the enzyme. So active site of the enzyme is the right site, where the correct site where substrate is going to bind. So answer is B for 6th question. Yes, Shreyas, you are right. Welcome to the class. Then 7th one. What A and B indicates in the given figure or in the reaction. So here I mentioned one reaction. What is this A and B? So for 7th one, it is C is the right option. A means enzyme and substrate. B refers to enzyme. P refers to product. So here we can observe the substrate, yes, combines with the enzyme. E, enzyme plus substrate, that gives enzyme substrate complex, that is what A. Then after the reaction it will going to give product and free enzyme. B refers to free enzyme. Okay, so right answer is C. You are right, Saurabh. Yes. Next question. Which of the following enzyme would correct glucose into glucose 6-phosphate? Which will going to convert or correct that glucose into glucose 6-phosphate? It is transferases. Transferases is the right answer. Just a minute. So transferases is the right answer. D is the right answer. Next, glucose plus ATP, that gives glucose 6-phosphate plus ADP. Which enzyme occur in this reaction? So here one enzyme is there. Which is that enzyme that is involved in that process? So 9th question, it is hexokinase. Yes, sir. For this one it is hexokinase. Okay, that will going to convert glucose into glucose 6-phosphate, and ATP is utilized, it is converted into ADP. Next, question number 10. Which enzyme would change glucose 6-phosphate into fructose 6-phosphate? It is isomerase. The reaction is called isomerization reaction. Glucose into fructose. Glucose 6-phosphate into fructose 6-phosphate. Who will going to change? That is the isomerase enzyme. Isomerization phenomenon. Next, question number 11. Which enzyme involved in hydrolysis of maltose to glucose? Maltose is converted into glucose plus glucose by which enzyme? So 11th one, synthetase, galactose, maltase, Lyases. That is the maltase, isn't it? So maltose gives glucose plus glucose in the presence of maltase enzyme. This is what? Carbohydrate, one of the carbohydrate, or we can say the digestion of biomolecule, that is maltose. Then the enzyme which also known as Caesar. So 12th one, it is Lyases. Lyases refers to, it will going to break down or cut, so it is called Caesars. Molecular seizure is the endonucleases. Okay, endonucleases, they are the restriction endonucleases. Restriction enzymes, they are called molecular seizures, isn't it? Yeah. So Lyases is the right answer here. So which enzyme is known as seizure? If you take molecular biology, then molecular seizure, it is the restriction enzymes. Okay. So right answer for this question number 12, it is A. Then 13th question. Which enzyme involved in conversion of a molecular into an isomer? Molecule to an isomer. Which is that enzyme? It is the isomerase. A molecular into an isomer, it is by isomerases. The question number 14. Here the enzyme which break up fructose 1,6 biphosphate into fructose phosphate known as what? Or enzyme involved in the conversion of fructose 1,6 biphosphate into fructose phosphate. 14th one, it is aldolase enzyme. Aldolase enzyme will convert this process. Then which enzyme is needed to digest food reserve in pea seed or garden pea plant? Garden pea seed. So which enzyme is needed? That is the protease. Why? Garden pea seed is rich with the protein. So protease is the enzyme that digests the protein particle. Yes, Saurabha Patil, for 14th one, A is the right answer. Okay, you are right. Then for 15th question, it is the protease is the right answer, because the garden pea plant is seed, plant pea seed is rich with protein. For that protein digesting enzyme, proteases will going to work. Yes, Manoj, welcome. Namaste. Next, the best example of extracellular enzyme. Best example of extracellular enzyme is, that is digestive enzyme. Why digestive enzyme is called extracellular? These enzymes will going to work outside of the cell in the gastrointestinal tract. So it is called extracellular enzyme. These enzymes are never going to work inside the cell. It is outside of the cell in the gastrointestinal tract or in the digestive tract. We can observe the reaction. Then a non-protein component of enzyme is called a non-protein component of enzyme is called what? Enzymes are generally made up of proteins. The non-protein component of the enzyme, it is called cofactor. Cofactor and a coenzyme, that gives holoenzyme, isn't it? Then which is an enzyme that joins acetic acid to CoA with the help of energy? In the plant physiology, here it is the Acetyl-CoA synthetase. Acetyl-CoA synthetase is the enzyme that will help in the joining of acetic acid to coenzyme A. Coenzyme A. So right answer is C for 18th one. Then 19th question. Okay, my dear students, if you have any doubt, you can ask. Okay, doubt idre ಕೇಳಬಹುದು. Next, 19th question. Let us have the discussion on this answer. Just a minute. Carbonic anhydrase is activated in the presence of which element? Carbonic anhydrase is the enzyme that is required with respect to that carbon dioxide transport in the case of human being under respiration. So which is that enzyme? So here, if you take the concept for the 19th one, it is D. Zinc Zn is required for that purpose. Okay, right answer is D for the question number 19. Yes, it is a zinc. Then question number 20. Vanadium is necessary for the activation of which enzyme? Vanadium particle is required for the activation of which enzyme? That is nitrogenase, for nitrogen fixing. Nitrogenase enzyme. 21st question. Find out the correct group of cofactor which is activated adenylase enzyme. So 21st, it is B. Magnesium, manganese and zinc. Okay. So for the 21st question, B is the right one. For 20th question, Saurav Patil, you are right. C is the right one. Then which one is not a coenzyme out of the following option? It is the ADP. ADP is the energy currency molecule, isn't it? NAD, NADP, FAD, ADP. Out of that, not a coenzyme. So coenzyme ಆಗಿ ವರ್ಕ್ ಆಗದೇ ಇರುವಂಥದ್ದು ಯಾವುದು? That is the ADP. Next, find out the correct group of enzyme. Correct group of enzymes which is activated by calcium particle. So 23rd, nitric oxide synthetase, protein phosphatase and adenylkinase is the enzymes that are going to be activated by calcium particle. Then which of the following set is not coenzyme? Is not coenzyme. That is NAD, FAD and ATP. Okay. Then coenzyme differs from prosthetic group because of what? Coenzyme differs from prosthetic group because they deactivates the enzyme. They do not attach it with the apoenzyme. They attach the apoenzyme with the loosely bound and they activates the enzyme. So 25th, it is C. They attached apoenzyme with the loosely bound. Okay. So because of this reason, coenzyme differs from prosthetic group. Then maltose is composed in which form, or maltose is made up of what? 26th question. It is made up of two glucose molecule, glucose plus glucose particle joined to form maltose. So because of that reason, when we are having the digestion of maltose by the maltase enzyme, that will provide two glucose molecules. Next, ribonucleoside formed from ribonucleotide. Why? So ribonucleotide is going to be there. So ribonucleoside formed from ribonucleotide. So hೀಗಾಗುತ್ತೆ. That is, it made from phosphate. So it made from phosphate, ribonucleotide, ribonucleoside. So when it is having the phosphate group attachment, it will going to have that phenomenon. 27th, C is the right answer. Okay. Next, question number 27 ಆಯ್ತಲ್ಲ. Okay. Yes, Saurav Patil, Manoj, Shreyas, Manohar, Sheshika, Saurabha, yes. If you have any doubt, you can ask. Okay. Next question. DNA is localized in which cell organelle? So DNA is localized in which cell organelle? DNA is going to be there in which cell organelle? Vacuoles, lysosomes, Golgi apparatus, then D, nucleus, mitochondria, chloroplast. So here we can observe the presence of their own DNA. So nucleus, of course, it is a central part of the cell, controlling or commanding center of the cell, which is with the DNA having the heritable character. Then mitochondria and chloroplast both are having their own DNA. So it is having their own DNA refers to mitochondrial DNA and chloroplast DNA. So ಎರಡು ಕೂಡ ಇರುತ್ತೆ. So chloroplast DNA in the chloroplast, mitochondrial DNA in the mitochondria. So that is circular type of DNA molecule will be there. In the nucleus, if you take eukaryotic cell, there the chromatin threads will be there that will going to form the chromosome under condensation phenomenon. So anyhow, this nucleus, mitochondria, chloroplast are with the DNA. So DNA is well common in the nucleus, in the eukaryotic cell or in the prokaryotic. Prokaryotic cell, nucleus is not the nucleoid, of course, but DNA is there. And in the mitochondria and chloroplast, there in their matrix, we can observe the presence of DNA. So it is located in the mitochondria and chloroplast. So cell organelle other than nucleus, the DNA is present in mitochondria and chloroplast. Okay. Next question. 29. So here I added about 30 questions. Yes, 28. D is the right answer. Sarav Patel, you are right. Okay. Next question number 29. See, a DNA strand is directly involved in the synthesis of all the following except, which is that one. 29th, it is D, the protein. Another DNA strand, that is a replication. tRNA production, mRNA production. So for these things, DNA is going to be directly involved, isn't it? Transcription. So transcription will give the RNA from DNA, and protein is from RNA to protein. So direct involvement of DNA is not there in the protein synthesis. It is indirectly involved in the form of mRNA. Okay. Next question. So question number 29, the right answer, it is D. Okay. If you have any doubt, you can ask. Okay, my dear students. And last question. Nucleic acids were discovered by whom? Who identified the nucleic acid in the cell? So Crick, Wilkinson's, Watson and Michel. So it is by whom? It is by Michel. Michel given the or discovered. Given means discovered the nucleic acid in the cell. That is a DNA and RNA nucleic acid. So that is what the total number of questions in today's class. So this is the last class from my side regarding with this year. And in the next year, means in the January 1st onwards, I am going to take next YouTube classes. So January 1st, January 1, we are having mock test on biology. Time is same 9 p.m. And on 3rd, I am going to take breathing and exchange of gases under human physiology, the next YouTube classes. And after that 3rd, there will be many more YouTube classes. Okay. So if you want to join, you can join to the plus subscription Unacademy. You have to do one thing. You have to download the app from Google Play Store, Unacademy Learners app. Install it and go to under search. Go to Karnataka CET and use my code, that is KCET Murali and get 10% off under this plus subscription. So we are focusing on Karnataka CET 2021 aspirants. Okay. Then comprehensive batch, the Mestros batch. It was started from 15th of December. You can enroll your name here. In the next month I am going to take human reproduction, reproductive health, and the sexual reproduction in flowering plant, followed by which that is the molecular biology topic. Okay, in the Mestros batch it is from 7 p.m. to 8 p.m. Then comprehensive batch, best plan for KCET aspirants, and multiple batches launching every month. So that is what our aim towards the 2021 KCET aspirants. So please join and prepare well for your oncoming KCET exam. So daily T20 free practice test also there. You can enroll your name for this test and champion batches. So that is the end of this class. Thank you everyone. See you again in the next class in the next oncoming year. So I wish you all the best. We will see in the next class in the next year, that is what 1st of January at 9 p.m. to discuss the mock test questions on biology, KCET biology. Thank you one and all. Good night to all. I am going to wind up this class now. Thank you.